I don't have a lot to cover today. Oh, let me get Janae in here. It's really thunderstorming here. Jess, Sia, you guys got any weather going on over there?
Not yet, but
Um,
it's gonna pick up soon.
yeah, ours will too. It's not raining yet, but it's supposed to pretty much all day.
Yeah, it was like we got a lot of rain. Like I was just looking out at the pool cover and it's like there's a lot out there from last night. So a lot going on there. Okay, so I graded everything this morning that I have so far. So both of the quizzes and your first labs are graded. Those neural labs, I see some of you have submitted them. Um, but if not, if you can get them to me by today or Monday, I'll probably try to grade those Monday. I know some people were still waiting on their packages. Sia, you got yours?
Yeah, I got mine and did it.
Okay, perfect. Um, Okay, so I just want to discuss the digestive and renal lab, and then we'll do a little bit of review from sensory phys. So, let's see. Hope this is the right one. So, for the digestive lab, there's not a whole lot that we'll be doing as a group. Glucose testing. So, I think, Janae, are you a subject on that one?
Um, I think I might be. I'm not for sure. I can't remember.
I think you are. And so there's a little video that I posted that's like how to use your lab equipment. And that'll show you exactly how to use the glucometer because you're going to do like some finger pricks to get um,
Oh, yeah, I am.
your blood glucose. So watch that video if you're not familiar. It's pretty simple. I didn't leave the instructions in the package, but it's pretty simple to use. So basically what Janae's going to do is she's going to get her fasting glucose. Again, you can do it before any meal. Probably easiest to do it first thing in the morning, like before you've eaten, but sometimes these labs, like during the school year or around lunchtime, so I'll have students come in fasted before they do lunch. And then you're going to, so you'll take your blood, you'll eat your meal, and then start a timer once you finish that meal. So 30 minutes post meal, 60 minutes post meal, 90 minutes post meal, you'll take those glucose levels again. Okay. And then each student in the class will be responsible for analyzing what that person is consuming. Okay. So Janae is going to tell you she had a bag of Doritos and a bagel or whatever it is. So then you'll look up, the nutrition data for each of those things. Okay. Describe in detail. Again, this doesn't have to be a formal like lab report or anything. Just make sure you cover each of these questions. So again, you don't need to write this assignment in like APA format for this first part. But describe in detail the carbohydrate digestion. So use what you'll have from lecture 4, which is today. So that digestive lecture. And that'll go through everything that you need right there. Okay. And then what does that glucose test measure? So again, remember we're looking at blood glucose. So it gives us an idea about how quickly and how much sugars we're getting into the blood after we've consumed it and absorbed it. Um, Again, methods of carbohydrate absorption, that'll come straight from the lecture as well. And then party requires you to kind of really think about it a little bit. Janae's the only person in our class that was doing this experiment, but I posted this morning for you um, 3 people from a previous class. Let's see if I can figure out how to get back to there. Um, okay, I'm going. Digestion, here we go. So I posted this morning just 3 people's data that I had. So here's their glucose at the top, and then the meals that they consumed. And then Janae will add her info too, so you'll have 4 people to kind of analyze for that. But again, thinking about qualitatively what they're consuming and what you know. Again, a lot of you have already taken a nutrition class, so that'll be helpful. But also after watching that digestive lecture, you'll have an idea about, okay, well, almonds have a lot of protein. Protein is slower to break down. So you'll have some more information about, how that might affect the speed at which we're going to see those nutrients show up in your blood. Let me go back. Okay. Questions on the glucose analysis? Okay, so now at the end here, it says type a summary of your findings, double-spaced APA format. So do I ask you to run a statistical analysis? Yes. So basically for this portion, so you're going to do a statistical analysis. What do you think would be the best statistical analysis for you to run? If I'm looking at fasted, 30 minutes post, 60 minutes post, 90 minutes post.
Repeated measures.
Repeated measures, ANOVA. Yep.
ANOVA.
Yep. So when you do your summary of your findings, you know, provide your results from that statistical analysis, and then you know, talk about what your p-value is and why it was significant and that sort of thing. Okay. Shouldn't be too crazy long. Then the second question is about metabolic syndrome. Again, you'll need to do some research to figure out what is metabolic syndrome and then how it affects each of these regions or each of these organs, I guess. And then you'll want to find one scholarly research article to kind of summarize the disease. Any questions on the digestive portion of this lab? Okay. The other thing that I'll kind of give you a heads up on, and we'll talk about this in more detail on Monday. How do I get back there? I wish you could just like see my screen all the time, you know? That'd be really neat. Okay. Here we go. All right, so the other part of this lab, again, this is kind of like a two-part deal, is lactate. And normally what we do for this is I would put one or two of you on a bike and we would actually measure your lactate threshold. We won't be able to do that because I can't have you in class or prick your fingers right now. So I gave you some mock class data, which is here. You'll have group presentations associated with that data. And again, I'll talk in a lot more detail about what that is on Monday. So we'll go over that information Monday, and then those presentations will be on Wednesday. All right. So I think that catches us up as it relates to lab stuff. Any questions on any of the lab material? How sensory stuff go okay? Looks like a lot of you got your sensory data already posted. That stuff went okay? 2-point discrimination went well? That makes sense, how to do that?
I was a little confused. Like, were we supposed to start at like the 8 and then work ourselves like?
There's not really like I mean, it's, you kind of just have to play around with it. Because like if you start at 8 for your finger, well, that's easy, you know? So it just depends on the area. And then you kind of want to try and trick your partner so that they don't, you know what I mean? So that they don't know what you're doing, I guess, if that makes sense. The video, I think the video was okay at explaining it. It's just, yeah, you kind of just got to try to trick your partner. Questions on those presentations. Those presentations will be on Monday as well. You've got your group assignments. Some of you are on your own. Some of you have a group. And you
Is there
Go ahead,
a specific time limit?
Nope. However long you need to discuss, you know, those different points. So, and you should be able to, if you had a partner and you don't know that person, you should be able to find them on our email list, you know, from those group emails that I've been sending out. So you can get in contact with them that way. So we'll start with that on Monday. Again, you can do a PowerPoint if you want, or however you guys want to do that. And then you can both talk, or you can audio record parts of it. If your partner can't be here, you can't be here, whatever. However you want to present your information is fine. Exam 1 on Tuesday. All right. There's a study guide posted. Again, my recommendation is to obviously watch all the audio lectures, take good notes. You've been doing that. You've already had 2 quizzes. So I would definitely do that, and then do the study guide. Okay, so once you've watched all those lectures, and the lectures go up to what you cover today, so the renal and digestive information that you cover today is the last part for that exam. Yeah, so do the study guide and make sure you understand those processes and things like that. There will be a few essay type questions, I think, if I remember right. Any questions on that? And again, you'll have time Monday if you do, as you work on this over the weekend. Everybody's good? Okay. So the last thing I want to do is just review. Let's start with hearing, and I want you to, Jess, you're our official, you have the touch screen, so you get to be our official note taker. I just want to review the process associated with hearing, and I also want to do the same with eyesight. So let's start with hearing. So if Jess can maybe pull up like a whiteboard, and we'll just kind of go through those steps associated with hearing, because I think that one's probably the hardest one of the senses. And we'll kind of go through how sound is transmitted. Okay, so I want you to maybe pull up your notes and think about, what are the structures that sound must travel through? What type of receptors do we need? Where in the brain is that information transmitted? Yeah, that's what we're doing. Let's do
You
it.
need to disable or
Oh,
maybe
yeah.
allow me to share my screen.
I wonder if there's a way that, there we go, that just like automatically happens. All right, crush it.
So do we talk about which is what happens first, second?
Yeah, maybe start with going through like the structures that it'll travel through. Maybe that would be good to start with, like the anatomy piece.
Mm. Well, I think sound waves, they go through like the acoustic meatus.
Am I supposed to draw these, or are we just like going to discuss it first and then?
It's your life, however you guys want to do it. Whatever is helpful for you.
Should we just go 1, 2, 3 like?
Wait, what's going on? Sorry, I had to go to the bathroom real quick, so I had to leave for a second.
So we're going through, Janae, we're going through the hearing. So we're going to start with like, the structures that the sound must travel through, and then talk about how hearing is transmitted.
Okay.
So review from your sensory lecture.
First, I think it goes into the external acoustic meatus.
It's like the outside part of your ear, right?
Yeah.
Okay, well, I'll try to draw this. Okay. Okay. Sound waves are going that way.
And then there's the eardrum.
The waves produce vibrations on the eardrum, which is also like the tympanic membrane, I think is, yeah.
That's like a bump here.
And 3 middle ear bones that those vibrations go through. I really don't know how it
We'll just do 3 B's for bone. There's 3 bones in here.
And then the vibrations reach the cochlear duct, which is where the inner ear, I think.
I don't know how to draw that. Pull up a little picture, see if I can draw one. Okay. Looks like we got some loops going on. There. That's what the picture says.
Like a snail, it looks good.
And then that vibration causes the stimulation of hair cells within the cochlea, I think.
So, are they like over this way, or is it somewhere like up here that it's like?
I'm not sure, but I can try to look it up.
Because it looks like there's some like nerves to the over here on this side.
Yeah. I'm not finding a very good picture. I'm really not finding a good picture, Jess. I'm trying. Um, I really can't find a good picture because I don't know.
Okay. Well, we're talking about like the nerves, right? Or is there something else that we're drawing?
it's just like the hairs inside the ear.
Oh yeah, the hair follicles inside the cochlea.
Is it in
And
this thing here? Yeah, okay.
sure. And then it just goes to the temporal lobe.
So that's just like,
So maybe like the brain, and then
there's our brain. Goes up there.
All right, now see if we can label some of these structures.
mm,
For the three bones, the malleus, incus, and stepes, but I don't know which bone is which. Um,
It looks like the malleus is like the top left one, and then the incus one is the top. I mean, you know what I'm trying to say.
This one? Or
Yeah.
do I have these all positioned wrong? I just threw them in there.
I think you do need to know the order through which they will travel.
so it goes the malleus, then the incus, and the stapes, because the stapes is closest to the semicircular canals. Like, can you see that? Those little white ones? Those are the three bones, if that helps at all. I don't know.
Wait, hold it up again.
Okay,
I
okay.
kept it really close to my screen.
Yeah, I'll let you see the there.
Did you draw that, Sia?
Cool.
Oh yes, I did.
What's the last one called?
The stapes.
All these wrong, but it's okay.
You're good. Yeah, because that one's right next to the semicircular canals.
Call that. Okay.
And then for the inner ear, the semicircular canal is the one that kind of looks like a snail.
Ooh, I don't know what I'm doing.
Oh, you zoomed in. Ooh,
It's zoomed in?
a little bit. Maybe. I don't know.
Oh. I mean, I meant to do that.
And then the outside ear is the external custos medius.
Or pinna, if you want to keep it easier.
What'd you say the easier option was?
Pinna, Pinna.
And then the tympanic membrane is what has the eardrum, or like is the eardrum in the little more inner of the ear.
And what was it called again?
The tympanic membrane.
Did we need to include the auditory tube?
Where's that at?
It's like the ears right there, and then the auditory tube is like the one that goes all the way in. I'm looking at the picture on the lecture.
Oh, we could definitely add that. Why not?
So it comes like right here, right?
Yeah, it comes like out there.
Okay, well, it's gonna go through the words because I'm not gonna move them all. Let's just come through here.
So a fun fact about that tube, that's where you get, like if you needed to get tubes in your ears when you were little, it's there. And it helps to drain fluid.
And what is it called?
Auditory tube.
The what?
Auditory
Okay.
tube.
Oh, that's horrible.
And then we can label the cochlea.
I really don't know how to spell any of these. That's okay.
Yeah, that's good enough. We get the idea. Okay, tell me about when sound waves become vibrations.
Here?
Yep, when it moves through which structure?
The eardrum?
Yep, so when it moves through the pinna and through the eardrum. So then what do the malleus, incus, and stapes do?
They help to amplify the sound vibrations. Wait, yes, right?
Yep. So as those waves transmit into vibrations, those three bones make it like stronger basically, so that we are more attuned to what's going on. Okay. What type of receptors do we find in the cochlea? Those hair cells, what type of receptors are they?
Mechanoreceptors?
Yep. So as those sound waves come into the cochlea, again, they're going to like shake the hair cells basically, because there's fluid in there. And so when those cells shake, then that's what stimulates the transmission of that signal from those hair cells then to the temporal lobe. How do you feel about hearing? What are the semicircular canals doing? What are they involved with?
Well, they're important for balance.
Do they play any role with the transmission of sound?
Mm. Do they collect the sound?
What do you think? What do you got in your notes?
Isn't the cochlea more important for the transmission of sound, though?
Yeah, those semicircular canals are going to be important for balance. Okay, so again, they're also fluid filled. So, like when you did that nystagmus test, so when you had somebody spin around, I know a couple of you have already done it, and then you watch to see their eyes tick. The reason their eyes are ticking is because that fluid is still circulating as that person is still trying to track their image, basically.
I
So,
thought that was so cool when we did that.
I know, isn't it
It's
cool?
weird. It's weird.
Yeah, it is. Um, yeah, so think of it like I don't even know what to use as an example.
Like a glass of water when you stop spinning.
Yeah, exactly. When you start swirling it and then you stop, it still swirls, right? So the momentum allows it to continue swirling, and that's kind of how those semicircular canals work. So, questions on sound? This one's like the messiest of them all, I think.
Good.
All right, let's go to eyesight then. Let's try eyesight, and that'll be our last one for today.
So the first pathway or first step would be like photoreceptor stimulation, which is different wavelengths of light stimulate the rods and cones within the eye.
So yeah, start with the external eye here. So what's got to happen in order to reach those photoreceptors? What do we got to pass through?
Pupil.
Yep. What else is up there? That helps with accommodation.
Mm. The ciliary muscles.
Yep.
Yeah.
What are they pulling on?
The lens.
Yep. Good. Okay, what's the fluid that goes behind the lens?
Uh, like, is it some kind of gel?
Yep.
Okay. Uh, I call the vitreous gel.
Yep.
Okay.
Yep.
What is it called?
The vitreous.
Vitreous humor, I believe. All right, so you got through the pupil, you got through the lens, you got through vitreous humor. Now we get to the photoreceptors.
Should we draw like the retina? Or
like the ganglia cells, and then bipolar cells, and then the rods and cones.
I would need pictures for reference.
Okay. Um, ooh,
We'll do this. Good old textbooks do.
Wow. Fancy. Next week she's gonna be adding like color and like glitter.
Yeah, no.
You'll have all the tricks figured out.
Do like 3 of these. Wow, here we go.
Yeah, buddy. Okay, so which one does which? You got cones and rods.
cones are responsible for color, and then the rods, like I think they detect the movement things.
Yep, so just remember cones, color. And if you had to choose like a section of your eye, where would the cones be more concentrated? The center or the peripheries?
The center.
Correct. Okay. So now we've went through the pupil, the lens, the vitreous humor. Now we're at the cones and the rods. So then what happens?
They go through like the bipolar cells,
Yep.
send the signal
And then,
to the ganglia cells.
yep. And then,
The optic ganglion crosses, or the optic chiasm or whatever happens with the optic ganglion cross paths to the opposite side of the brain.
correct. Okay, so we have that chiasm or that cross. And then,
To the occipital lobe.
what was that?
Does it go to the occipital lobe after that?
Yep, the occipital lobe. I couldn't hear what you said. Yep.
Does it go from there? Does it go to the occipital lobe?
Correct. Yep. Do you feel like eyesight's a little bit more clear than than the hearing? The pathway seems a little more like, I don't know. I think besides the crossing thing, otherwise it's a little more clear, I feel like. Um, okay, questions on eyes. Questions on any of the other senses? We didn't spend a whole lot of time with like taste, partially because taste isn't like totally well understood. Or many of the other senses. Any questions on any of those?
I have a question about the eye.
Yeah.
It's like doesn't happen often, but sometimes people's eyes like come out. You know what I'm saying? Like they pop out of their socket a little bit in the front.
Like,
How does that happen?
um,
Like do the muscles there just like not work and it's like comes out? Or, like how does that happen?
you mean like they fall out, or what do you mean?
No, like I had this friend at school, she'd like push on her eye like on the side like this and it would like come out of her head.
It
Ew.
has to do with well, a couple things. So like your bony arrangement, some people's eyes are going to be sunken in more than
Mm-hmm.
others, just simply based on like the thickness of some of these bones. But then
Mm-hmm.
also just like any like ligament in your body, like some of you are more flexible than others because your ligaments are more lax than others. So if I had lax ligaments in my eyes, and remember there's like 1,000, that was an exaggeration, there's like a bunch of ligaments that are keeping your eye in its location, and also a bunch of little intrinsic muscles. So if those were loose, then it would be like easier to do that.
Mm-hmm.
Also, this is another weird thing. Like if I had a lot of like fat over my like eyelids, that would be harder to do, right? So
Mm-hmm.
somebody that had, I don't know what causes that or anything, but like if you had, like thicker eyelids, then that wouldn't happen. I don't know. I don't know.
Okay.
She's got loose ligaments.
Okay.
I've never seen that before, besides like a cartoon.
Really? It's weird.
Makes me nervous. She needs to wear goggles.
She probably should. She doesn't do it often though.
Other questions on any sensory stuff? Okay, questions on digestion that you'll be for that lab that you'll be working on. Okay, I think that's all I have for today then, peeps. Um, and like I said, Monday we'll have sensory presentations. Monday you have a quiz on the digestive system. And then on Monday we'll also cover a little bit more info about that metabolism lab. So I have a very short PowerPoint and I'll kind of explain, how we would have done that lab. All right. Okay. Have a great weekend. Let me know if you need anything. Otherwise, I hope to see you on Monday.
All right, thank you. Bye.
Bye,
Thank
everyone.
Bye.
you.
Bye.
Yeah.